In this module, we will discuss another important method to quantify the protein. And the method is isotope coded affinity tags, ICATs. And an isotope coded affinity tag, ITAG, that is as the name indicates, is an isotopic labeling method that is used to quantify or that is used for quantitative proteomics by mass spectrometry that uses chemical labeling agent. You see, in this method, the tags, they are used by chemical reagents and they tag the different peptides, different protein molecules, and give us the weight or quantify those molecules. Now, what experimental do? They couple affinity tag to protein through the sulfhydryl groups of their cysteine side chain. You see the sulfhydryl group of the amino acid, cysteine, the side chain which are present, they tag them with the different proteins and affinity tags which are made. Now, these affinity tags, they typically contain 3 parts. Affinity tag, they are consists of or made of 3 parts. For example, in this figure, a generic affinity tag is shown, where affinity region, that is a biotin, which is present, then the middle portion, which is there, and it attaches to the sulfhydryl reactive side or sulfhydryl group of a cysteine amino acid. That is a generic ICAD tag which is attached. Now, how does this help to quantification? How does this tag help to quantify the protein, to quantify the mass of the protein? So, just consider a cell grown under 2 conditions. One is with and without serum. That is one, the cells, they are grown with the serum, and the other one, they are grown without the serum. Then the question is, how much change we see in the concentration of proteins when serum is added to the medium in which the cells are grown. When the cells are grown, what is the difference in the quantity? Because now this is a part of the quantitative method for that, quantitative proteomics. Just consider this, that using ICATs to measure the change in the protein concentration upon shift in the growth condition. Now, the growth condition is when is present, when is grown in the presence of the serum, and another without serum. So, you can see that one is the condition 1, which is a light tag, and then the condition 2, which is a heavy tag. Now, they are attached to the protein, and then what is happened, that once that protein is attached with a ICAT thing, it is hydrolyzed by the hydrolytic enzymes. Then the affinity purity tags, they are attached with it, and with it, this whole mass is determined by LC mass spectrometry. And you can see that there is the retention time, which gives us that what is the weight of the protein, what is the quantity of the protein or the peptide, and the second one is that how much it is present, which is on the y-axis. That is what is its absorbance, what's its concentration, which is present over there. Now, in this case, the investigators or scientists, they could add light ICATs to the protein from the cell grown in absence of serum. That was the condition 1, which I mentioned. And heavy ICATs, which are heavier molecules, to the protein from the cells grown in the presence of serum, condition 2. So those are the two, how they are added. Now the protein could be mixed. So these proteins, they could be mixed, peptide mix, hydrolyzed with proteolytic enzyme, a protease such as trypsin, affinity profiled, purified using the affinity reagent that they are purified into the different components, and then they are later subjected to LCMS, that is the liquid chromatography mass spectrophotometry, that spectrometry. LCMS, which is the liquid chromatography mass spectrometry, which they are subject to that. And then what happened? The heavier of the two peaks in each pair comes from the cell grown in the presence of serum. That is, in the two conditions, as we see, the cells which are grown in the presence of serum and the cells which are grown in the absence of serum, where we get the heavier peaks in the LCMS, and that is with the presence of the serum, because the serum provides more energy, provides more proteins which are there, and the lighter of the two comes from the cells grown without serum. So the cells which are grown without serum, that is the lighter peaks, the lower peaks which we get, and this helps us to differentiate between the two proteins or between the two components of the protein which are grown there, and help scientists to distinguish the weight and to determine their mass which is present there, to quantify the different proteins.